Hello listeners, welcome to a special edition of the InnCoCells Podcast. This is recorded in Krakow, where the InnCoCells project is attending the International Congress on Natural Products Research, along with two of its sister projects, that's Secreted and Marbles. We're actually jointly presenting a breakout session of the conference. It's entitled Prospecting Aquatic and Terrestrial Natural Biological Resources for Biologically Active Compounds. So that does sound like a bit of a mouthful, but basically the background to it is that InnCoCells is one of 4 projects funded under the same Horizon call. And what that means is that we've basically got very broadly similar aims. We just focus them in slightly different directions. So the 4 projects are called Algae for IBD, InnCoCells, Marbles and Secreted. And the first letters of those 4 projects happen to be AIM&S. So it comes out with a very handy acronym, AIMS. So that's what we've called ourselves. And representing the AIMS cluster, I'm delighted to welcome 2 special guests today. We've got Dr. Manuel Salvador, who is the coordinator of the Secreted project, and Dr. Fernando Reis from the Marbles project. Thank you both very much for taking time out of the busy schedules, because you both had presentations today. So thanks for joining me in the InnCoCells podcast. I think it would be a good start. I'll turn to you first, Manuel. If you could tell me a little bit about the Secreted project and how you came to be involved in it.
Hello, so I'm Dr. Manuel Salvador, and my background is mainly in systems and synthetic biology. So all my career, let's say that I was learning a lot about bacterial metabolism and how to engineer those to overproduce certain biotechnological products. And I was that lucky that when I wanted to come back to Spain, I found this company that is IDNR. I joined them, and then they are like devoted to the development of research projects. So we were thinking what was the kind of topics that fit me best. It was a matter of, let's say, calling back my friends and then make like, do you join me? And I think also that whenever I later explain what the project Secreted is about, then I think that we have like something different regarding the other approaches, because in our case, it was more about reutilizing already collected things, already data that is available. And that I think that was why we are like the 4th option, like a risky, let's say, strategy. Not a risky, because it's something that makes a lot of sense, but it's not like the classical approach where you are like addressing new natural sources, looking at them, how we can, let's say, exploit them to get a new compound. And in our case, it was like, okay, that happened before. Let's see what we can reutilize from the things that has been already collected.
So you're talking about repurposing things which have already been discovered, finding new applications for them.
Yeah, and that has a lot to do with the term sustainable, because we think many times that the sustainability happens at the later stage when you try to make the approach sustainable because you are consuming raw feedstocks or waste in converting into biotechnological products. But I think that the sustainability happens transversally along the whole project, and it's starting from the very beginning when you are not going through the environment and sample from there, affecting and altering the ecosystems. And you have done that in the past. And there is a lot to do with those magnificent resources that are already collected in many microbial collections, that in some cases, they are forming part of these so-called negative results that are in many cases not that negative, because they have something to offer.
Yeah, so for our listeners who aren't specialists in this field, just to give some context to what Manuel was saying there, sometimes, or often actually, I think mostly would be fair, experiments don't work. And you end up with data which basically says this doesn't work, which is fine, but what it's actually saying is it doesn't work for the purpose for which it was tested. It doesn't necessarily mean it's not useful in all contexts. So where you have a collection of 1,000 bacterial species, say, which don't do what you want them to do, doesn't mean those species don't do anything useful. They may be useful for somebody else. And this is the basis of what's known as a strain collection. This is like a library almost of organisms. And you can repurpose that library by testing them for something that wasn't on the original list of objectives. So this is basically what the Secreted project is doing. And what are you looking for?
We are looking for biosurfactants and siderophores.
Right, okay, so that is a bit of a mouthful. Briefly explain what both of those are.
So we are working and we are all the time in our daily use. We have a set of compounds that are what we call, let's say, soaps and detergents. So anytime that you wash your hands, you are working with a family of surfactants. So basically, from a chemical point of view, those are molecules that they have a part that is hydrophilic and the part that is hydrophobic. So the hydrophilic ones, the hydrophilic part, it permits that you can utilize that with water and then you can wash your hands. And the hydrophobic part is the one that get the dirtiness out of your hands. So a part of being detergents, and there are many different detergents in the industry that are utilized for many chemical industry purposes that, I mean, it's crazy. I'm not getting deeper into that. We have the ability of these molecules to organize themselves. That is the most interesting application, that how they can organize themselves into these micelles. So for the audience, the micelles is anytime that you see an advertisement of a cosmetic and then they have with a new nano encapsulation and you can see these little balls, how they penetrate into the skin. So those are these kind of micelles that can be used to surround any other molecule that has the application.
Oh, so like a carrier of some kind?
It is a carrier.
Yeah, okay.
That's it. I was looking for the word. So in those terms, that is why we have in the European Union, this biosurfactants is one of the 5 topics with the highest interest in biotechnology applications. And because they work as carriers, they solubilize things, they can act as detergents, and many other secondary applications that are coming out of this, let's say, technology tree. And also, just trying to divert the route of application through siderophores, some of these biosurfactants, they say they have the ability to chelate metals. So the chelation is that they trap, they interact electronically, let's say like a magnet. And it is because bacteria normally, when they are in nature, they need, of course, some metals to act for their enzymes and for surviving. And the iron is very insoluble in nature, so they had to secrete some molecules that are catching them, and then by means of how they are coming back, they can, let's say, utilize that to feed themselves with irons for the different purposes. So some of these biosurfactants has been described as they have the ability to chelate iron. Interestingly, some siderophores in the marine ones that are when they're in the sea, they are also amphiphilic. So we have a interesting field where we have, let's say, it's not a black or white, there is a gray area. And that also portrays a different range of applications that are related to how we can chelate metal. So it's the metal recovery for the mining industry, also for the reutilization of metals that are coming from the chips that we normally work with.
So this would be like, they say now that the rare earth metals are becoming very difficult to find in nature. So you're talking about using siderophores and biosurfactants to reclaim precious metals, if you like, from consumer goods.
And also for the daily based metals, because when you have the mining industry, the application of siderophores is increasing the production yields. So it's in the normal, let's say, and the very rare metals. So it's for both of them. And also because of the bacteria capturing iron using these siderophores, you can utilize them as what is called the Trojan horse. And we have spoken before about the problem that we have with antibiotics. So this could be a new source of antibiotics because you can utilize these molecules to trick the bacteria to catch these molecules that has linked another molecule that can be poison for them.
Oh, that's very interesting. So basically, the bacteria are going after the metal, but you've attached an antibiotic to the metal, and therefore they take that up at the same time and it destroys them, but they don't know that at the time they're going after the metal, so it's basically fooling them into basically killing themselves.
Yes.
Interesting.
Because actually, in terms of the bacteria, when they are in consortiums too, they are living together, and that evolutionarily that happens that there are some bacteria that are producing the siderophores and some others that are taking advantage of them. So those are the cheaters. So in terms of evolution, there are many bacteria that are sharing the transports for utilizing this, because if I can take advantage of another one, so that make it possible that we can utilize these siderophores to take advantage of that cheating system to portray a new antibiotic.
That's really interesting. So what we're saying here is that the research isn't focused on a specific application. You can use it for water purification, you can use it for metal recovery, you can use it for making a new generation of, or not necessarily a new generation of antibiotics, but overcoming the fact that antibiotics don't work by making bacteria take them up. Presumably you can also use it for like remediation of agricultural land, that kind of thing, by collecting metals from the soil. So what's your path to commercialisation? Have you got companies in your consortium who will take the products forward, or is that another step after the project?
We have what I call the divergent nodes into, let's say, the stakeholder network. So what I mean with that is that we have, for example, Sfera. Sfera is a spin-off of the University of Verona that started by analyzing how they can encapsulate different compounds, and they ended up to have the whole network of clients that they assess several biosurfactants and synthetic surfactants for them. So meaning with that anytime that Sfera is confirming the positive outcome of a biosurfactant, once we have handled the IPR issues, we can already, let's say, put that thing for their customers, that they are also another key players into the industry of surfactant. We have also Acuplex, the only European company that I could find that are experts on siderophores, and actually they have developed a very sensitive siderophore detection kit. And as well, they are working for the health industry in terms of trying to characterize whenever you have a pathogen in your biological samples because of the detection of the siderophore that belongs to that pathogen. But they have also a very interesting network within the health industry regarding the conjugates of antibiotics to the siderophores. So anytime that we have an interesting outcome, we have the key players that can advertise that within their clients.
That's very similar actually to InnCoCells. So we've got, it's like a concentric circles. We've got the companies and academic partners who are doing the bio prospecting, and the ones who are then seeing if they can be scaled up. So it will be interesting in some way to the industry. Then we've got the ones who are extracting it and the ones who are testing it. But some of those people who are testing it, the companies also are marketing cosmetics. So they're getting things which they can then take forward themselves if they want to. But outside that, we've got a stakeholder group where we have the larger cosmetic companies like La Prairie and Clariant, who could take over a really successful cosmetic ingredient and provide the way to commercialise it in a big sense. So it seems like we've got a fairly similar strategy for moving products out, if you like. Fernando, how about you tell us a little bit about yourself and how you came to be working in the Marbles project?
So my name is Fernando. As you said, I come from Fundación Medina in Spain, who is an institution dedicated to do mainly microbial natural products research. And we were part of the consortium of the Marbles consortiums, where we are trying to develop new antimicrobial compounds to treat the infections related to plant diseases, fish diseases, and human health.
Okay, how big is the consortium? How many partners is there?
We are 14 partners coming from 10 different countries.
Okay, similar to InnCoCells, we've got 17 from, I think, 11 countries. Okay, so that's interesting. What are the objectives of the Marbles project? What are you trying to do?
We want to harness the potential of marine microbes to produce new products related to treat diseases in plants, in fishes, and also in human health. And we want to find new strains, new microbial marine strains that produce molecules interesting in those fields.
So essentially, if I understand correctly, you are screening marine resources, looking for bioactive molecules, and the application of those molecules will be in animal health and human health. Any particular area of human health, or what sort of indications?
Mainly as antimicrobials, because there is an urgent need to discover new antibiotics, but we also might find some products useful in cosmetics and in other areas.
Well, that's good, because obviously that's what we do. We can probably share those results. Just for some context for our listeners, so why are antibiotics important? Well, it's because there's very few in the development pipeline, and a lot of the ones we have got are not so good anymore at what they used to do. Lots of resistant species emerging, so we need new antibiotics which have different mechanisms of action to try to get further along in this. It's kind of like an arms race. We have to outpace the evolution of bacteria with our drug development. So in terms of impact then, your project, you are hoping that you will come up with some new pharmaceutical compounds which will be clinically valuable. You'll be able to use them actually to treat people and to treat veterinary diseases.
Yeah, of course, that's the goal, but also it's important to find greener solutions for plant diseases and for fish diseases, because it's important that compounds used to treat plants and to treat plants infections are not harmful to the environment and to the, I would say.
So sustainability then is equally important to efficacy. This is, again, very similar to the InnCoCells project. I mean, of course, we're going to have similarities, but it's striking because we have this exactly the same thing. We want to have products which work as on the skin, topical application, antimicrobial, anti-inflammatory, etc. But they have to be sustainable. And also, as I'm sure is important in your project, you have to look carefully at issues like the Nagoya Protocol to make sure you're not taking genetic resources without due consideration for their origins.
Yeah, of course, we are aware of the Nagoya Protocol. Actually, there is a work package within the project dedicated to define best practices regarding the collection of samples and the sustainable use and the access and benefit sharing with all the communities involved in the generation of the samples.
We're talking in the context of the project. How far can you take this development pathway? So, for instance, in InnCoCells, we have the aim to, we call it, bring the ingredients to a pre-commercial stage. So we're not actually going to end up selling anything in the project during the duration of the project, but we want to get them to a stage where they are ready to be taken on by a stakeholder or a commercial partner. Is that a similar kind of?
It's pretty the same situation in marbles. We want to develop products. By the end of the project, our goal is just to have a scale-up method to produce the most interesting molecules that we identify within the development of the project.
Yeah. So let's have an example. When you talk about marine organisms, are you mostly talking about microbes, or are we talking about seaweed, or do you have a broad spectrum of species you're targeting, or is it focused on one particular type?
It's mainly microbes. Isolated from different sources. Like, for instance, we are isolating strains from the skin of salmon, just trying to combat infections in salmon with those strains. And we are isolating also microbes from sponges, from algae, and from all kind of marine
Fantastic.
organisms.
That's really interesting, because I somehow had formed the impression that if you're screening for marine microbes, you're just basically looking in the water, but no, you're looking actually on other organisms as well. That's very interesting. And of course, if they live there, they are probably fulfilling some kind of purpose beneficial for that organism, maybe protecting it from pathogens. So it would be a natural, logical development to exploit their protective molecules, whatever they're making. That makes perfect sense.
That's okay. And we are also exploring consortia of different microbes, because microbes are not alone in nature. So sometimes the effect that you're looking for is not produced by a single strain, by a group of strains. So we're trying to develop assays, and we are trying to develop a methodology to study the consortia of marine microbes that might be producing molecules of interest for.
That's a very interesting point, because I keep on returning to this over and over again in our project. Take a plant and we make an extract of it. We put it on someone's skin and it does something nice. So we think, great, find out what's causing that. And the scientist's mind is to try and break it down and find an individual ingredient. But what we've mostly found is the individual ingredients don't do as much as the extract. So the conclusion is that you've got lots of things working together rather than having a single ingredient with the major effect. And of course, in nature, that's going to be the case, isn't it? Because nature doesn't work, like you said, with single things. It's a community, lots of things working together. So the benefits and the effects are, if you like, overwhelmingly likely to be caused by communities of organisms and communities of molecules rather than by single high-impact ingredients. So yes, that's something we've been looking at as well in the context of finding naturally produced plant-derived molecules which either protect the skin from microbes or from inflammation or do whatever, keep your skin hydrated, all those kind of things. I'm going to turn now, if you don't mind, to the reason why we're all here. To remind the listeners, we're here at the conference because we're jointly presenting a spin-out session, a breakaway session. And the idea of setting up the AIMS cluster was, I think, I can divulge this, it was Secreted's idea. We got approached by you, and you also must have approached the other partners at the same time, with the aim of finding some commonalities which we could pursue. So my personal experience as a dissemination lead, it's been very handy because we've shared, we've made joint brochures, and we've done joint social media campaigns, etc. But from a scientific perspective, how have you found the interactions? Has it been helpful?
Yes, it's very interesting how we have addressed this topic, this horizon call. But if I think as a project manager, I also think that in the points that where we overlap, but we don't overlap in terms we share, we have many possibilities because we can share also the stakeholders. In many cases, the stakeholder network that we have is being, let's say, as a consequence of what our respective contacts. And I would like also to consider the possibility that our respective stakeholder network can review our respective results and to see further collaboration schemes. So I think that the AIMS cluster is a very good idea in those terms.
Yes, so I think this is also very much encouraged by the Horizon program itself. From what I remember, we have to make a commitment to open science, which includes what's known as fair data sharing. So again, for the benefit of our listeners, fair in this case means findable, accessible, interoperable, and reusable. And the last one is probably the most important in the context. So all 4 projects will generate an awful lot of data. Can it be reused in other projects to get more information from the data and more useful products, processes, etc.? And yes, I would really think, because we've all put a lot of effort into the work we've done, if we can get added value from that for future projects, that would be an excellent way to move forward. Yeah, a great legacy for all 4 projects to do. Fernando, just want to ask, how do you characterise those kind of activities in the AIMS cluster from your perspective?
Well, it's good to know what the other projects are doing. For instance, in the project Secreted, they are trying to develop a new cytosine. Cytosines have many properties, and who knows, maybe one of the compounds that we eventually identify as bioactive might be a cytosine. We might find something in common too.
Yeah. Well, plants produce cytosines too, so you never know. We might all be working towards the same thing. That's true. Yeah. Well, thank you very much, both of you, for taking the time to come to the podcast recording. It's been really fascinating talking to you. So thank you both very much for coming. Thanks to our listeners for tuning in, and we'll see you on the next edition of the InnCoCells Podcast later on. Thank you very much, and goodbye.